Okay, are there any questions? Let me state by saying we're going to be covering Lab 01, the ubiquity of microorganisms. You should remember that your online plagiarism quiz is due 11:59 p.m. this Saturday. You must achieve 100% on this plagiarism quiz, or else you'll get 0 points on the infectious disease project, and it's worth 100 points. You also need to read the infectious disease project. There's a file called the project instructions on Canvas for next week. And I actually need to tell you a little bit about that. And I meant to show you in the lecture that I had done something to the lab website, and I didn't show you in the lecture, so let me show you now. Are there any questions before we get started? If not, let me share my screen. All right, so you're seeing now, can everybody see this?
Yeah.
Somebody verify.
Yes.
Thank you. So now you're seeing the general course information. And let me go to an announcement. I just made one today because a student asked a very good question. What do you do to get a good grade in this class? And I listed down 6 items that I've noticed in other students who get a good grade. So take a look at that if you're interested in getting a good grade. There are other announcements here that you should look at. One, Clark College Resource of the Week. They actually want me to discuss it each week, which I'm not going to do, but I've discovered the easiest thing is now to just put a link here, and they will add to it. They have 2 links up now for week 1 and week 2, even though week 2 hasn't started yet. So if you're interested in some of the things Clark offers, take a look at the resource of the week. The virtual support center is open on campus. I'm not sure what that's about up there. The emergency grant funding is available to Clark students. And week 1 was very light. I'm a little bit surprised it was so light, but let's go back and look at week 2. Penguin Pantry. They have a pantry open at Clark, and you can pick up monthly food boxes through registered students. And you can read about that here. So if you're needing food, for example, and I don't know what the requirement is, but you can take a look at that. And like I said, if you're needing food, go and pick it up. I don't think you have to sign up for it, but you do have to come in and pick it up. And then the activities calendar and student success workshops, students with disabilities luncheon. Anyways, that will be each week, and you have to look at it each week because there will be a new link posted each week. Any question about that? You guys have been at Clark, so you should have seen Clark College Resource of the Week before, correct? You've seen that before,
Yes.
haven't you?
Yes, we have.
Okay, great. All right, let's go back to my Clark main page, because there's something I was supposed to state in the lecture and I forgot. Reminder, first, this lab assignment to lab 00, worksheet for 00, it has to be turned in by Friday at something like 8 a.m. because on Friday, I have to turn in to Clark College students who signed up for the class and then never showed up. And so This will be my only time for dropping students. And if you don't have worksheet lab 00 turned in, you will be dropped. So make sure you turn it in by Friday at 7 p.m. Most of you have already turned it in, but if you haven't, it needs to be turned in by then, or you will be dropped. This is your last warning. And then, of course, you will have to turn in worksheet lab 1 on Saturday. The normal time we turn everything in, 11:59 p.m. on Saturday. The other thing I wanted to mention is, I've already mentioned, but I just want to reiterate, there's an extra credit assignment. Turn in your picture for extra credit, and I forgot how many points, 3 or 4 points. The extra credit study sheet, this one has to be turned in by this Saturday. And I think this one is worth 6 points of extra credit. And then next week, there's another extra credit assignment. And you need to get together and form a study group with another student, a microbiology student. And you can use your cell phone, you can use Zoom. The Zoom that you're using now, you can use to get together with other students for forming a study group. So I don't need to do anything. And as long as you only have a few number of students, I don't remember how many, you can do it without having a Zoom account. But if you need to, you can use the Zoom account that you're using for my class. And obviously you can't use it during class time, but you can use it for your study session. Anyways, to get the extra credit study point, starting in week 2 through something like week 9 or 10, each week you can get together and study. It needs to be for at least a half an hour. Submit a document to collect the point. You must include your name and the full name of everyone in your group. Now, the good news is only one person in the group has to submit it. But if everyone submits it, that's okay. But only one person has to submit it. Tell me what you studied, when you studied, the date, what you studied, and how long you studied. Okay.
So we have a big group chat going for everybody, except for like maybe 1 or 2 people. Would that count as a group study, or is that too many people?
What are you doing in this group chat?
Well, right now we're just talking about the classes and whatnot, but it's just so far we have everybody's name for the group chat.
Well, that's fine if you do that to study. You cannot use this extra credit study assignment to work on a current assignment, especially the lab. And I've got a warning here. You may not study this week's lab and receive credit for the lab as well as extra credit for your study session. You have to do the lab work and the lab time. It's not for making extra credit study time. And I never had a problem except for last term. Students were continuously doing that. Okay. For some reason, we're stopping the share. I don't know what's the case. Let me start the share again. So if you get together on a current assignment and work on that assignment, first of all, that's not studying, and that's not going to be for extra credit because you're getting points for that assignment. All right. So I want you to study, but you don't have to. You can, I mean, I want you to quiz yourselves, but you don't have to. It does have to be a study session where you guys are studying the material that you will be quizzed on, or there will be an exam question on. Okay. Ideally, you will be studying and quizzing each other. And you really shouldn't do it with everybody in the class, because if you do it that way, and you even get together for an hour, you're not going to be having time to quiz everybody. You should break it up into smaller groups of, I don't know, 3 or 4 people, 5 people maximum. so that everybody can be quizzed and everybody can study. The idea of quizzing each other is usually if a student notices that another student is weak on a topic, they will continuously quiz that topic. And then that student will become better versed in that topic. You understand? That's what I want. Anyways, to get the extra credit, it's 1 point for each week. You need to include the full names of everyone in the study group. You need to study something you will be quizzed on. You need to tell me when you did it because you're getting the points for that week and for how long you studied. It has to be a half an hour or longer. And then submit it to the document. You can make it, submit a file, or you can just type in all of this information in the page. Any questions about that? You get 1 point per week, so you can't wait until the end and then submit 9. You can only submit 1 per week, but you can get at least 9 points, maybe 10 points on it. Do you guys see this? The stop share keeps flashing. I'm not sure why that's the case.
We see it.
Yeah, we see it.
All right, great. All right. I think that's all I meant to talk about in the lecture. But I've covered it now, so you're good. Any questions about the class? All right, if not, you can get the lab module, which we're going to be talking about module 1 here, and the worksheet for lab 1 down here. I'm not going to go there because I've got a special one. And let me remind you, you need to read the instructions for the infectious disease research project. And I'm going to close that now. You can find that under week 2 lab, infectious disease research project. You open that up, it's the instructions. And it's 100 points. It's not due until February 19th, but I'm going to talk about it next week. So I want you to look at it. And you will begin working next week on this project. Any question about that? All right. So that's all I needed to discuss. Let's go ahead and talk about the lab project. Let me see if the lecture is finalized. It has, good. I meant to do that before class started and I forgot to look. Hopefully I got it done because last time I wasn't able to get it done, but they didn't seem to bother starting the class even though it was still formatting. So that's good. Let me see if I've got it here. Oh, I've got 2 of them here. Why is that? Oh, this is the one I want. I've got it highlighted for one I want to cover. All right, lab module 1, ubiquity of microorganisms. There is some reading you should do in the textbook to help you with the lab module. And that's in the pre-read, page 157 to 162, and then 162 to 163. There are some video clips in this lab module. You should watch them. Your learning objectives, you should know that microorganisms are almost everywhere. Be able to describe the purpose of culture media. Be able to describe the terms colony abundance, colony diversity, and colony morphology. Be able to define the terms inoculation, ubiquity of microorganisms, sterile, isolation, incubation, in vitro, culture media, and there can be different types. There can be a broth or an agar, The auger is usually put in a plate, but it could be on a slant or a deep in a test tube. There are fastidious organisms. There's non-selective media, and that's the only one we're going to talk about today. And then there are ubiquitous organisms. And then tell me what a colony is. Any questions about any of that? All right, microorganisms are organisms that are too small to be seen with the naked eye, meaning without magnification. It does include microorganisms, meaning cells that are too small to see with the naked eye and they have a cell, as well as viruses and prions. And we'll talk about viruses and prions later in the term, although I did briefly go over viruses in the lecture today. We will also include helminths, the worms in a microorganisms, because as we discussed earlier in the lecture, helminths can cause human disease. They can be pathogenic, and their infective stages is usually, actually, I think the infective stages is always microscopic. The phrase ubiquity of microorganisms refers to the concept that microorganisms are everywhere, or essentially everywhere on the planet. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. You'll also learn some basic lab protocols, including the use of solid media, labeling lab cultures, procedures for disinfecting the lab benches, and procedures for washing your hands. We'll talk about culture medium. The culture medium is also called a growth medium. And in a microbiology lab, it will be a sterile laboratory preparation, meaning sterile, it has no living organisms in it, that contains a variety of nutrients that are required for growing microorganisms. Culture media may be liquid, and these are called broths. So you can call it a liquid media or a broth, and they're commonly used in a test tube. They can be made solid by adding agar or something else to solidify it, in which case they are grown usually in a petri dish or a plate, same thing, but they can be a solid media in a test tube. Agar is a gelatinous substance extracted from seaweed, and it's just used to solidify a liquid media and turn it into a solid. It has a gelatin-like property. All right, you need to watch the video on preparing pour plates, so that's right here. And then this is an important question, but it may not be asked in your lab worksheet, but you should be able to answer it after you watch the video on preparing plates. Why must a culture media be sterile prior to being used? And we're talking about in a microbiology lab.
Because of contamination.
Yeah, the reason is you don't want contamination. And so it has to be sterile to begin with. Showing you different culture media. Here is a test tube with the liquid media, and then a test tube with a solid media that's similar. I mean, it's a deep, and there's a slant where the agar is in the shape of a slant there. And these are agar plates. Today, we're only going to talk about one media, a general purpose media. It's non-selective in function, meaning it does not select for certain organisms to grow. It's non-selective. It's usually of complex composition, and they're used to support the growth of many non-fastidious organisms. meaning many organisms can grow on a general purpose media. And they use the peptone, the sugar, and the yeast and the beef extracts to grow. Some examples of a general purpose media are nutrient agar with yeast extract, or NAY, and we will talk about NAY media a lot in this class. But we also talk about tryptic soy agar, or TSA, another general purpose media. And like I said, they're just cheap and a lot of organisms grow on them. And so that's why we use them. Nutrient agar is the cheapest, and it's like NAY, it just doesn't have the yeast extract added to it. It's nutrient agar, or NA. Okay, and we just use it to grow a lot of things. Now, only the non-fastidious organisms can grow in a general purpose media. If it's a fastidious organism, it will not grow, or at least not grow well, on a general purpose media, because a fastidious organism requires additives before it will grow. And we'll talk about that later, probably later in the term. You need to label everything you grow, and for you guys, you're going to be virtually growing things. So every tube and every plate, you label it with your name, the date, the type of medium, and if it's nutrient agar with yeast extract, you just put NAY on it. So there's somebody's name. That's actually an instructor. E. coli is the inoculum type of inoculum right there. And then the incubation temperature is optional. And then the date right there. TSA, the media, and this person's put the temperature that they grew it at. Any question about that? So everything should have your name, the date, the type of media, and the inoculum, meaning what you're growing. If you don't know what it is you're growing, you can label it for what you did, like exposure to air. So you just say air exposure, or soil. You got some soil and put it on the plate. You should label things by putting a piece of tape on a test tube, and you can write on the tape and then put the tape on the tube. On the plate, you should write on the bottom of the plate, on the periphery, so that it doesn't interfere with your seeing the growth in the majority of the plate. Any question about that? Hopefully I didn't move that. And why you label on the bottom of the plate is because that's where the microorganism is to grow. If for some reason you separate the top from the bottom of the plate, you have the bottom of the plate labeled for what this is. And that's where your microorganisms are. There was a lab technician in a lab I was once working in, and she had a whole tray full of different plates from an experiment that somebody ran. It might have been her, it might have been the instructor. And she was walking with this tray of plates, and then she tripped, spilling all of the plates onto the floor, and the bottoms and the lids got separated. And she had labeled everything on the top. And so the whole experiment had to be tossed because nobody knew what it was on the plate that they had. So label on the bottom. Any question about that? On the bottom of the plate. All right. Inoculation is when you place a microbial specimen into a culture media. You can put it in with a variety of tools, and we'll talk about that later. Incubation is the placement of the culture media that's been inoculated into appropriate physical environment. Usually in a microbiology lab, you put things in an incubator to grow them, and the incubator will warm them as well as provide some humidity. You can have more fancy incubators that provide a specific environment, like how much oxygen, how much carbon dioxide. I don't know what else they do, but they can do different things. But in our microbiology lab, it just gives it the air. The incubator can give it humidity, and it obviously warms the culture so it'll grow faster. When you grow agar plates, let me show you from my, no, I don't have a good, oh, here we go. Something to show you. Can you guys see this? I'm not seeing anything. Why am I not seeing anything? I should see. I should see. I should see a little icon of what I'm seeing, and I can't see anything here. That's weird. Anyways, I can't see it, but hopefully you can see this. I'll make it in front of me and then you're able to see. So when you put a plate in upside down, and this being the lid, the moisture will come out of the agar and then will condense on the bottom of the lid. And then it'll form a droplet, which will then drop onto the plate and then will disperse what you have on the plate. So if you have colonies growing on the plate, it will disperse the colonies. Any question about that? It may not contaminate the plate because inside the plate, it's sterile other than what you inoculated, and the moisture is sterile, and the dropping of the stuff should come from the lid, which is sterile. So it may not contaminate the plate, although on the edge of the lid, it's not sterile. And if the moisture touches the edge and then drops down into the plate, it could contaminate the plate. So it increases your risk of contamination, but it ruins the culture because it spreads the growth in the culture. Any questions about any of that? Sorry, I'm not seeing what I'm supposed to be talking about, so I'm having to look where it is and bring it back. There we go. Anyways, because of that, we never inoculate, well, we never put plates into the incubator right side up because the moisture will condense on the lid and then drop down into the plate, potentially ruining the plate. What we do is we put the plate in upside down in the incubator. Now, if moisture forms in the plate, it will form on the agar, and the agar will wick it throughout the agar so that the agar will never drip down onto the lid, which it really doesn't matter if it drips onto the lid because you don't have any growth on the lid. Any questions about any of that? So everything you grow in the microbiology lab in a plate, the plate should be put in the incubator upside down. And that's to prevent moisture or condensation from forming and then dripping on your culture. Any questions about that? All right, I need to talk to you about a bacterial colony. Bacteria colonies tend to be rather smoothly textured on their edge and their surfaces, but there are exceptions. And a fungal colony tends to be furry or hairy. Once again, are exceptions. So when you usually look at colonies, you can distinguish, are they bacterial or are they fungal? In this lab, we won't be growing fungal colonies. So if you see a fungal colony, it's probably from contamination. A colony is between 10 to 100 million cells. And all of them might have been descended from one cell or descended from a very few cells that were placed on the agar. And then with time, though that one cell or those few cells grew into the colony that you see. So you can't see one cell on the plate, but you can see a colony, which has at least 1 million, maybe 10 or more million cells on it. And how those cells grow, the bacteria cells grow mainly by binary fission. We have a little video on binary fission right here. So you can watch a link on binary fission. This is an incubator. And it looks like this one has a few more controls than we have. But there, that's providing humidity for the organisms. And you then shut the door and it heats to do that. But that's a typical incubator you find in microbiology. Here is a fungal colony, and you can see it looks fuzzy or hairy. The same with that one. Let me blow that up a little. And that looks different. No, that's kind of blurry, so let me shrink it down. Hairy or fungal looking. And this is a bacteria colony, which has a distinct edge, not necessarily round, like that one's not real round. These aren't round when you put them together. That one's not round, but it doesn't look hairy. Okay. Any question about that? All right, let's talk a little bit more about colony characteristics. Colony characteristics are shown in this table here that I'm showing you. And the good news is you don't need to know most of these terms. I will provide this graph if I ever want you to distinguish between, I don't know, rhizoid and filamentous in shape. You might know the common ones like circular and irregular. You should know a normal colony margin, we call it entire. That's like a round one. And then these are the others. I would never ask you to distinguish between lobate and undulate because they're so similar. And I never ask you, is this a rhizoid or a filamentous? But I might ask you, is it undulate or rhizoid? So you should be able to distinguish between those two, and obviously entire. If you look at the colony from an edge, you can determine, is the colony flat, raised in a mesa? Is it convex or is it umbunate? This is mountain shape. I won't ever ask you, is it pulvinate or convex? Because that's too difficult even for me to distinguish between those two. The pulvinate is more extreme convex. We could ask you, is it punctiform? And a good way to say, is it punctiform? Is it smaller? I don't know if you can see this, the pen prick or the pencil prick, it's punctiform if it's smaller than that. Small is about the size of a pen or a pencil. Moderate is in between the pencil size or the pen size and the eraser. And then it's larger than an eraser on a pencil. Any question about any of that? You can say the colony is smooth or rough, and you have to look at it to determine that. Hopefully, if you look at that colony, this one right here, you'll see that's smooth. This one is rough. And actually, students have the hardest time trying to distinguish smooth and rough. And it is a little bit subjective. I probably in an online class won't even ever ask you, is the colony smooth or rough? But I will ask you colony characteristics, meaning where you have to describe a colony characteristic And you do that, I think, in this lab. You can say whether the colony is glistening, that means it's shiny or dull. Let's come back here. So that's a shiny colony. And that one is a dull colony. This one is also dull. Lastly, you can say whether the colony is pigmented or not, and whether what its optical properties are. The optical properties, if you don't see any light coming through, it's opaque, and most colonies are opaque. I think all of the colonies on this plate are opaque, with the exception possibly being the edge of this one. In which case it might be translucent. I'd have to look at that closer to see, is that opaque or is that translucent? I don't know. Anyways, translucent, you can see through it, but it's like seeing through a muddy water. You can see through it, but you can't see perfectly through it. And then translucent is you can see right through it. Very few colonies are translucent. You know, that one there, it's really small. It could be, excuse me, transparent is where you can see right through it. This might be translucent right there. Let me show you another example. And then you can see at least opaque from translucent. Let me shrink that down. Move faster, come on. There we go. All right. This one is opaque. That one is translucent. And I don't see any in here that are transparent. Any questions about that? Pigmented is whether the colony has color or not. If you see a color, it is pigmented. If it's non-pigmented, there is no color, and that could be cream colored, white, off-white in color. It's coming down here. This one is pigmented. It's kind of a brown color. That one's kind of a pink color. You know, there aren't many colored ones in here. Let's go up here. There's a yellowish one and I don't know, brown yellow or dark yellow. And that one's white, non-pigmented. This one is kind of gray, non-pigmented, or you could call that off-white. Any questions about the colony characteristics? All right, so figure 6 is showing you some, oops, wow, well, that didn't work well. I did something wrong. There we go. Come on. I might have to stop this over. What's the layout maybe?
I think you just need to zoom in more.
Say again?
I think you may just need to zoom in more. You're at 50%
No,
zoomed in.
I've got several pages being shown. I only want to show one page at a time. And I don't know how it did that. Let's see if that does it. No, that's still not doing it. Nobody knows how to do that? Yeah. I'm not seeing how to do it. Let me just close this down. I'm going to reopen it because I'm not sure what I did. I'm not there anymore. think it's this one. Ah, same problem. All right, we're just gonna have to deal with it.
So if you go to the bottom right-hand corner, there's a little bar. And if you put that over more, because it's only at 50%, if you click on
I'll
that
blow
little,
it up, but I only want to show one page at a time.
yeah. So right now it's at 50%. You need it to be at 100% in order to show one page. So if you move that little bar over, it should help.
Right, okay,
Awesome.
thank you. Now, where were we? Oh, here we are. Showing you the different colonies, take a look at that. When we're talking about abundance, let me blow that up so I can see it better. Abundance is simply the total number of colonies on the plate. And I'm not going to actually count it because there's too many here to count. But the abundance is someplace between 30 and 60, maybe even 70 on this plate. And you need to count them to get the true exact abundance. I'm just going to say between 30 and 70. Two colonies that look different from one another probably are different species. Let's blow that up really big. That's good for the next question. Like this colony looks different from that colony. So these are probably different species. That one looks different. Hey, there's one that's translucent. So these are 1, 2, 3 colonies right here. And this one is probably different because it's so different in size. So 1, 2, 3, 4, 5. That one's probably different, so that's 6. This one is clearly different because it's different in color. That one is even different because it's different in color. And you can use different characteristics to figure out different colonies. Any question about that? You need to know different colonies, meaning different species, for answering the diversity. The diversity is the number of different kinds of colonies on an agar plate. And you have to look at the colony characteristics to identify the total number of diversity. This one, it has too many colonies for me to even do it. But if we come up here to this plate, we can say, I'll blow that up a little bit. These two are probably different in fungal and different from all the other. And then these are different from everyone that's not yellow and round, but it could be the same as all of these. So there's 1, 2, 3, 4, because that one's different. 5, because that one's a small, tiny white one, although that could be the same as that one. So this one has something, and there's 6. I didn't count that one. So this plate has at least 6 different colony types. So the diversity would be at least 6. 1, 2, 3, 4, 5, 6. Okay, so at least 6 will be the diversity. Anything less than 5, I'd say, is probably not correct. And anything more than, I don't know, maybe 10 would also be incorrect. So you have to look at it to determine the diversity, and then you have to go on the colony characteristics. Now this probably is the same, but you could argue that that's a much bigger colony than that one. So you could argue that those are different. But in my case, I'm going to say that those look pretty similar, that I'm going to say they might be the same. But if you look really, really closely here, it's possible that this one is different than that one, because that one looks slightly darker than that one. I'm not certain those are different, because colonies can have slightly different characteristics. What would be a good example? Like this one and that one are different sizes, but they may be the same colony. Okay. And this one and that one are different colonies, but they have a lot of the same characteristics. They're both shiny, they're both entire in their margin, they're both similar color, pigmentation, both opaque, etc. So those are probably the same colonies. All right, let's go back down. For getting the diversity, it does take a little bit of interpretation. The abundance, though, you just need to count the number on the plate. So for this example here, what's the abundance of cats in this picture? Come on, count your cats.
7.
1, 2, 3, 4, 5, 6, 7. So the abundance is
7.
7. And what's the diversity of cat species?
2.
2. You have lions here and cheetahs there. There's a question you can answer, but it's not required for the lab. Okay, so you know the terms in this lab. And then complete the laboratory assignment. Let me go get that. I don't have it here. So answer the questions in the Word file and submit it by 11:59 p.m. Saturday of this week. And protocol 1, we're looking at a plate that was exposed to the air, and you in theory just left it out on the air and then looked at it. And this is what's in the air you breathe, and this is the plate here. And then answer these questions. What is the colony abundance on this plate? You need to count them. Colony diversity on the plate. If you're 1 or 2 off from what I think it should be, it won't be wrong. But like on the colony diversity here, if you say something like 20 different diversity or 3 different diversity, I'll say the answer is wrong, okay? Meaning I've got a number here and you have to be at least close to my number. And you could always argue that, I don't know, this colony looks different from all others. That colony you could argue is different from that one by size, although that one I would say may not be different. But that one is different from that one, etc. Anyways, do the count for the abundance. That's just the number. And then the number of diversity, different species on the plate. Now the abundance sometimes is a little tricky. Like right here, is that a colony? Because that sure is right there. That is not, but this right here is. But is that a colony? And this one I would argue may or may not be a colony. It might be just a satellite of one of these colonies, probably the big one here, but maybe of that one. Okay. So I will also allow 1 or 2 off on your total abundance number. But that's a different colony, that's a different colony, that's a different colony. Another thing is that, let me blow this up. Is that a different colony inside of this one? And you see, this is sort of yellowish and funny shaped right there. Or is it a part of this colony? And that one there clearly is a different colony than this one here. And I just know it's different. This one here, I don't think that's a different colony. It's not distinct. The edge is not distinct. All right, any questions about any of that? Anyways, that's procedure 1. Procedure 2 is what gets on your arm when you put your arms on the bench. So what you're doing here is you're going to inoculate the bench before you clean the bench and then after you clean the bench, and then make a comparison. And here we have a swab of the bench before you clean it and then after you clean it. And the first question is, did table disinfection work? And what you should do in answering these questions is P2 or procedure 2, question A. Okay. Did table disinfection work? Obviously, yes. What's the purpose of allowing the table disinfectant to air dry? You can actually find that in this lab module if you don't know, but it's pretty easy to get your answer on that one. Procedure 3, what's on your hands? What you're doing is you're touching the plate before you wash your hands, and that's shown here. And then you're washing your hands and then touching the plate, meaning you're not touching with your hands anything else before you touch your plate. And this is touching what you have on your hands after you wash your hands. And this is what you have on your hands before you wash your hands. Oh, excuse me. You never have to answer the first question, which is the procedure, what's on your hands. Because that's just telling you what this project is about. There's a question. So procedure 3, the first question is, does hand washing work? And it depends what you define work to be. If that looks like it works to you, say yes. If you're saying that it doesn't work unless you remove all microbes, if that's your definition, then it didn't work. But you need to qualify what you mean by work. And then 2, give a suggestion for why hand washing did not result in complete removal of microbes from your hand. And if you don't know the answer to that, you can look online. There's a hint here that it may have something to do with the normal microbiota on your skin. And if you obtain your answer following research, provide a brief citation of the information source. Procedure 4, when you kiss your significant other, what exactly are you giving him or her? You don't need to answer that question. But we're going to have 2 label plates. One is going to be exposed to your lips, where you kiss the plate right here and right here. And the other plate, you're going to touch it with your tongue. So right there, somebody touched the plate with their tongue. Please note on the tongue plate that the lips, you can actually see the colonies, although they're quite many in here, small, and they're touching, but not all of them are touching. On the tongue, you'll note right here, if I can blow that up, that this smear here is actually a smear of many colonies that are almost essentially touching each other. And then there's a few larger colonies on the tongue, or bacteria that form large colonies on the tongue. The majority of the colonies are tiny, and that's why you have a smear. Any questions about that? Oh, all right. So which plate has higher colony abundance, your lips or your tongue? And why do you think that's the case? So answer B. And what might cause the observance and difference in the colony abundance between the lips and the tongue? C, why was the tongue plate incubated at a higher temperature than the lip plate? So when you look at the procedure here, we're inoculating the lip plate at 30 degrees and the tongue plate at 37 degrees Celsius. If you don't know, 37 is 98.6. And 30 degrees, I don't know what temperature that is in Fahrenheit, but it's above 72. It's probably above 75. Okay. So my guess is it's something like 80 to 85%. 80 to 80 degrees Fahrenheit. And then answer which plate is higher colony abundance. Why do you think it's the case? And then lastly, answer why was the tongue plate incubated at a higher temperature than the lip plate? Any question about that? Procedure 5 is looking at microorganisms in soil. What we do is we get a small inoculant of soil, put it in some water, and then take that water and streak it over the plate. And here we get the growth of the plate after it's been exposed to water that had soil put into it. And then answer, what is the colony abundance? Is it even possible to accurately answer this question? And then procedure 5B, is the greater abundance in colonies from the soil or the lip plate? Any question about any of that? All right, if you're all good, start working on the worksheet and turn it in by Saturday. I will be here until 8:15 to answer any questions, although I may get up and move a little bit. I might even grab a bite of dinner. All right. Go ahead and work on your lab.